These are the four major biomolecules of life.
A monomer is a small molecule. A polymer is many monomers joined together forming a repeated long chain.
Polymerization is the process of monomers forming long complex chain polymers. Below are the two different types of polymerization.
Carbohydrates are long chains of simple sugars called saccharides. All carbohydrates contain carbon, hydrogen, and oxygen. They all have the same chemical formula but have a different chemical structure. This is called an isomer compound. The formula for carbohydrate is C6H12O6. Monomer for a carbohydrate is called a monosaccharide. When two monosaccharides join together, they form a disaccharide. When many monosaccharides join together, they form a polysaccharide. The process that joins them together is called the condensation reaction. Monosaccharides and disaccharides are soluble in water, making it easy to dissolve the sugar. Polysaccharides are insoluble in water, therefore they can be used as energy storage. A condensation reaction involves two molecules reacting together to form a new larger molecule. When the reaction occurs, the molecules lose a hydrogen bond to form a covalent bond. This is called a glycosidic bond. Hydrolysis is the reverse reaction to condensation reaction. The large molecules break down into two small molecules, meaning the hydrogen bond is added to break them apart. Carbohydrates are important to the human body and the function of cells. They are used as energy and energy storage. Monosaccharides are simple sugars such as glucose, fructose, and galactose. Glucose is important to the function of cells because the organelle mitochondria use glucose and oxygen to produce ATP energy to power the cell. Glucose can be structured as an alpha glucose or a beta glucose. Disaccharides are sugars such as maltose, sucrose, and lactose. Here is a table showing the disaccharide, the enzyme which breaks it down, and the two monosaccharides that are formed after hydrolysis. The glucoses that form maltose are both alpha glucoses. The three most important polysaccharides of carbohydrates are starch, glycogen, and cellulose. Polysaccharides are great for energy storage as they are large, complex, and insoluble. Starch is a mixture of two polysaccharides known as amylose and amylopectin. The enzyme amylase breaks down the polysaccharides into maltose.
Lipids are a group of substances that are made up of mostly carbon, hydrogen, and oxygen. They are long chains of fatty acid monomers. Fatty acids are long hydrocarbon chains with a carboxyl group at one end. Hydrocarbon chains are long networks of carbon and hydrogen covalently bonded to each other. A carboxyl group is composed of a carbon atom double bonded to oxygen and singly bonded to a hydroxyl group, which in this case is oxygen and hydrogen. Saturated and unsaturated fatty acids are two different types of fats based on their chemical structure and properties. Saturated fats have straight and unbranched carbon chains where all the carbon atoms in the chain are bonded to the maximum number of hydrogen atoms. There are no double bonds between carbon atoms, which makes the chain straight. Saturated fats are solid at room temperature. Their function is to provide energy and protection to our body and vital organs. They also support regulating our body temperature. Unsaturated fats have a similar structure to saturated fats in the sense that the carbon chains are attached to hydrogen. However, in unsaturated fats, at least one or more carbon atoms are double bonded. This creates a bend or a kink in the chain. The double bonds to carbon mean there aren't as many atoms linked to hydrogen, causing them to not be fully saturated. Unsaturated fats are usually liquid at room temperature. Their function is to provide energy and protection, but they are also known to be the good or healthy type of fat which improves cholesterol. Unsaturated fats contain a double carbon bond in the middle of their chains. There are two different types of unsaturated fats, cis fats and trans fats. It is their arrangement that differentiates them both. In cis fats, the double bond has hydrogen atoms on the same side as the bond, creating a kink in the chain. In trans fats, the double bond has hydrogen atoms on opposite sides of the double bond, creating a straighter chain. Here are the four main types of lipids. Lipids are mostly hydrophobic, meaning they are resistant to water and therefore don't dissolve. An example of this is when we mix oil with water. The oil remains separate as the oil molecules are attracted to the oil and the water molecules are attracted to the water. However, glycolipids and phospholipids are known to be amphipathic, meaning they can be soluble in water. This is due to them having a hydrophilic head and a hydrophobic tail. Triglycerides are the most common type of lipid. They are made up of one glycerol molecule which is covalently bonded to 3 fatty acid molecules. Triglycerides are made by the condensation between the glycerol and a fatty acid, and they form an ester bond. They can break down into fatty acids and glycerol when the energy is needed. The opposite of this is hydrolysis reaction, where lipids are broken down and called derived fats. An ester bond is when the glycerol is bonded to each fatty acid chain by a carbon-oxygen-carbon bond. Triglycerides can then be broken down into fatty acids and glycerides by a process called hydrolysis. These molecules can then pass through the plasma membrane of the cell. They provide a long-term energy storage, protection to our body, insulation, and also store vitamin A and vitamin D. Phospholipids are similar to triglycerides, however, they only have 2 fatty acids attached. One fatty acid is replaced with a phosphate head, and the other 2 fatty acids act as tails. They are soluble in water, and this is how they form a double-layered membrane known as a phospholipid bilayer. The tails form the interior away from fluid, and the heads make the exterior, which is in contact with fluid. This makes it a semi-permeable membrane and allows substances in and out of the cell. The function of phospholipids is to protect and maintain the integrity and stability of cells, and to control the passage of substances, allowing the cells to control what enters and exits. Steroids are a subgroup of lipids, and they have several essential functions in the body, such as lipid metabolism, hormone regulation, energy storage, and anti-inflammatory properties. 2 of the most common steroids are cholesterol and hormones.
Proteins are large and complex molecules that have many important roles within the body. They are critical to most of the work done by cells and are required for the structure, function, and the regulation of the body's tissues and organs. To put it simply, proteins are large and complicated polymer molecules. Proteins are polymers. To be exact, they are called polypeptides, formed from a sequence of amino acids. These are linked together to form a polypeptide chain. Every amino acid is connected to the next through a peptide bond. This is created during the protein biosynthesis process, where cells make proteins. All proteins have the chemical elements carbon, hydrogen, oxygen, and nitrogen, and they often sometimes contain sulfur. The length of a protein can vary from thousands of amino acids to just a few. Proteins are an essential part of a cell's membrane. So what exactly do they do within your body? Proteins are extremely important to your body's functions. For example, hemoglobin is a protein that carries oxygen through your body. Your body relies on oxygen, therefore these proteins are needed in order to function in everyday life. Antibodies that destroy and attack microorganisms that are not welcome in the body are also proteins. Without these, we wouldn't be able to fight disease and illnesses off. Every enzyme inside your body is a protein. These are just a few examples of how important proteins are. Without them, we would not survive. Protein catabolism is a process where proteins are broken down into amino acids. A single amino acid monomer may also be called a residue, which is a repeating unit of a polymer. What you may not have known is that out of the 8 essential amino acids, 3 of them are BCAAs, which means branched chain amino acids. The 8 essential amino acids are phenylalanine, valine, threonine, methionine, leucine, isoleucine, lysine, and histidine, each of which plays an important role in your body. Although your body can make non-essential amino acids, it cannot make essential amino acids, so you have to get them from your diet. There are several non-essential amino acids which you only need during illness, pregnancy, infancy, or trauma. A peptide bond, a dipeptide, and a polypeptide are all essential keywords when talking about proteins. Protein complexity. There are 4 levels of protein complexity, and they are: primary, a sequence of amino acids linked together to form a protein; secondary, a region of amino acids that are stabilized by hydrogen bonds from the polypeptide backbone; tertiary, a polypeptide shape is formed into a complex molecular shape caused by R groups such as ionic bonding and hydrogen bonding; quaternary, there are several protein chains or subunits made of peptides. Each has its own primary, secondary, tertiary structures held together by hydrogen bonds.
Enzymes are molecules made from large proteins, around 1,000 to be exact. Enzymes are responsible for building large molecules or breaking them into smaller ones. This is known as a biological catalyst. Biological catalysts help speed up a reaction, remaining unchanged at the end of the process. Enzymes that can build large molecules are known as anabolic, and enzymes which can break large molecules into smaller ones are called catabolic. Enzymes can work intracellular and extracellular. Can you remember the organelle lysosomes? Lysosomes contain enzymes which are responsible for breaking down biomolecules, foreign materials, and apoptosis. Enzymes work all over the body. You can find them in your saliva, digestive system, and liver. They help with cell regulation, hormone production, and transportation. Enzymes also help with DNA replication. Enzymes work with one molecule in particular due to their quaternary structure. Here is a diagram showing the enzyme substrate complex. This diagram shows how the substrate binds to a specific enzyme, converting it into products. Here are the key words and a description of their function in the enzyme substrate complex. Here are the 6 classes of enzymes. These categories help identify enzymes based on their function around the body. Here are a few examples of some of the enzymes found in our body. Lipase help digest fats in our gut. You may be familiar with the enzyme lactase. Lactase helps break down the sugar lactose into glucose and galactose. If you have a lactose intolerance, this means you do not have enough lactase enzymes to help break down the sugar. You can find the enzyme helicase in DNA replication. Helicase unravels the hydrogen bonds that are holding the base pairs of DNA together. This happens during mitosis when genetic information is copied. A certain amount of energy needs to be supplied for a chemical reaction to occur. This energy is called activation energy. Enzymes are used to help lower the amount of activation energy required for a chemical reaction to occur. This means activation energy can work in lower temperatures by working alongside enzymes. Without enzymes, the chemical reactions will take longer and will need to use more activation energy.
There are 2 different concepts that describe how an enzyme interacts with their substrate. Below I have listed the 2 models. The main differences between the 2 models is that the induced fit believe there is flexibility in the interaction between the enzyme and substrate, whereas the lock and key model implies a rigid, precise, and perfect interaction between the active site and the substrate. Enzymes are sensitive to temperature and this affects their activity. In low temperatures, enzymes have less kinetic energy, resulting in fewer collisions, and this means it will be rare for the enzyme and substrate to bind. As temperature increases, the enzymes accelerate, resulting in more frequent collisions. Each enzyme has an optimum temperature it works at its best. However, if the temperature goes above the optimum, the activity of the enzyme rapidly decreases, and the enzyme loses its shapes, becoming non-functional, and this results in the cell being denatured. The pH level of the environment impacts enzyme activity. Enzymes have an optimum pH level where they function at their best. This is normally around pH 7. If the pH level changes, so does the charge and conformation of their active sites, meaning less activity. At extreme pH values, enzymes can become denatured. Enzymes not function at their optimum causes the protein to unfold, so the shape changes, meaning the substrate no longer fits the active site, so the reaction will no longer take place. Enzyme activity is also affected by substrate concentration. As the concentration increases, so does the reaction. At low substrate concentration, enzymes won't be operating at full capacity due to there being less molecules to interact with. As substrate concentration increases, so does enzyme activity, because there's more opportunities for the active site to bind. However, it's important to note that if the concentration kept increasing, the enzyme will be working constantly and at a fast pace, meaning there will be too many enzymes for active sites. This is known as Vmax.